Previously on Ball Z, our heroes discovered the difference between benign and malignant tumors. They also learned the different risk factors that increase the likelihood of developing a disease. But just when they thought they understood non-communicable diseases, they were attacked by the effects of smoking and diet. But by answering the various challenge questions that came their way, they were able to defeat their foes. And now, our heroes have found themselves in the jungle of plant tissues. Will they be able to describe the different organs and tissues of plants, or will they leave empty-handed? Find out in today's exciting episode of Ball Z. Here's the retrieval questions for this lesson. You know what to do. Benign and malignant tumors are the two types of tumors that you can have. Tar causes lung cancer. J.J. Thomson proposed the plum pudding model. Ernest Rutherford proposed the nuclear model. Viruses cannot be treated with antibiotics, and malaria is caused by a protist. I've already covered photosynthesis and the photosynthesis practical in this video, so check that one out if you haven't seen that already. Just like humans, plants have tissues and organs as well. To make sure we are all clear on these definitions, match up these different definitions that you see here. A cell is the basic unit for life. That just means that nothing smaller than a cell can be considered living. For example, a muscle cell. Tissues are similar specialized cells working together to perform a specific task, following the same pattern that would be a muscle tissue. Organs are different tissues working together to carry out a function. For example, you have your heart, which is an organ made up of nerve and muscle tissue. The pacemaker cells located in the right atrium send electrical impulses to your heart tissues, causing them to contract. Finally, organ systems are a collection of organs and tissues that carry out a specific functions. This example is the circulatory system, which pumps blood through 3 different types of blood vessels around your body and around your lungs. Click the card to check that video out if you haven't seen it. Here's an average plant. We have roots, stem, leaves. The roots anchor the plant into the soil and also absorbs water and nutrients via diffusion, osmosis, and active transport. The stem holds the plant upright and allows the movement of substances up and down the plant. Finally, the leaves are used for photosynthesis and also allows transpiration to occur. Before we look at transpiration and translocation, let's look at the specialized cells and tissues within plants. Here are 3 specialized cells that we have in plants. We've got the root hair cells, xylem, and phloem. Try and match up these with the functions that you think they perform inside a plant. Root hair cells in the roots absorb water and minerals. The xylem transports water and minerals up the plant. And finally, the phloem transports dissolved sugars around the plant. Root hair cells look slightly different compared to your regular plant cells. Oh yeah, this plant cell, the standard one that you see at the bottom here, these are technically called palisade cells. Now I have these challenge questions for you to have a go at. Do your thing. The root hairs help the root's hair cells absorb water and minerals because they have a large surface area. They cannot photosynthesize because they do not have any chloroplast, And this makes sense because the roots are located where there's no sunlight. This also means that they need to get their energy from the glucose that's produced from the leaves. Now let's see if you can match up these adaptations of a root hair cell. The large surface area provides more area for the substances to move in and out of the cell. The large permanent vacuole holds the cell in shape and speeds up osmosis due to the concentration gradient created by the cell sap. This means that the mitochondria are used to provide energy for active transport. This is how they absorb most of the mineral ions. You have bare minerals in the root hair cells and a few minerals in the soil, so diffusion wouldn't cut it. If you did diffusion, you will lose your ions and minerals, so they need to use energy from aerobic respiration to move minerals against the concentration gradients. You're moving them into the plant. We're gonna do the same thing now for the palisade cells. Match them up. Chloroplast obviously is the site of photosynthesis. Being located at the top of the leaf means that you maximize the amount of sunlight that hits you, and the permanent vacuole is used to hold the cell in shape. Now let's look at the xylem tissue. The main features are these hollow tubes and these lignin spirals. Lignum is actually Latin for wood, so that may help you understand the use of them. And of course, I have some challenge questions for you to have a go at. The barks are obviously made up of lignin.
Nice.
Looking at the xylem tissue, we see no mitochondria or any other cell structure for that matter. Xylem tissues are actually dead cells. They're made up of dead cells. For the last question, we have to remember that xylem is used to move water, and this is a passive process, so no energy is needed because water moving via transpiration, which we're going to look at in more detail, that is a passive process. Remember that the adaptations of xylem is to move water and minerals. This whole process is the transpiration stream, which we will look at in a bit. Use that and the previous activity to match these boxes up. The hollow tube provides more space for the movement of water. Being dead cells means that they do not need energy for their job, so that sweet glucose can be used elsewhere in the plant. They're being efficient. The lignin spirals allows them to withstand the high water pressure that can build up inside plants. Xylem tissues move water passively, so it simply just needs to move the water from the roots all the way up in one stream. But phloem tissue need to move the dissolved sugars made from the leaves to all the cells in the plant. So they have two main adaptations to help them with this. It can't be passive like the xylem tissue. This one will be a little bit trickier, but have a go trying to match these up. The sieve plates direct dissolved sugars using active transport. This means that the sugars can move against the concentration gradient, and that energy for the active transport is provided by these companion cells. It isn't shown here, but the vacuole in the phloem contains cell sap, which has the dissolved sugars, and this is what the sieve plates is directing around the place, so they're moving that cell sap in the vacuole. Let's now look at the structure of these plant organs, the leaves. Starting from the top, we have the upper epidermal tissue. This reduces the loss of water through the leaves. Then we have the mesophyll, which just means middle leaf. You can probably guess then what chlorophyll must mean. Well, since chlorine is a green gas, chlorophyll means green leaf. We have two segments in this middle layer, this mesolayer, this middle leaf. The one closest to the top is the palisade layer, as we saw earlier, that has bare chloroplast inside it. They're specialized to perform photosynthesis. And the fact that they're located just below the epidermal layer means that this photosynthesis can be as efficient as possible. Below the palisade layer, we have the spongy layer. So cool because it has these air spaces that just look like sponges. This is the equivalent to the alveoli in animals. It is the site of gas exchange, and so it allows the diffusion of gases. Now at the bottom we have the lower epidermis, which contains these tiny gaps called stomata, or stoma if you're just talking about one. If you're wondering why stoma kind of sounds like stomach, that's not an accident. They both share a root word for mouth, which is in Greek. The stomata allow gases to move between the plant and the surroundings. But this movement has to be regulated. Nate Dogg and Warren G had to regulate. This is the function of these guard cells here. They regulate the diffusion of gases in and out of the leaf by opening and closing. The final plant structure you need to be able to talk about are meristem, but I covered them in the differentiation lesson, so click the card to skip over there. I reckon the best way to cover the next part of the syllabus would be to look at a past paper question, but let's just first define transpiration and translocation. Transpiration is the loss of water vapor through the stomata, and as you can see, that's just one direction from the root all the way to the leaves in one single stream. Translocation, on the other hand, is the movement of dissolved sugars from the leaves to the rest of the plant, and this GIF we can see that the phloem tissues move substances in both directions, up and down, because all the cells, all the living cells inside the plant need that glucose to survive. So let's look at this past paper question. A student used a potometer to investigate the rate of water uptake in a plant shoot. Water is absorbed from the bottom of the shoot and is lost through the leaves. The movement of the water causes an air bubble to move towards where the water is being lost. And if you place a ruler here, you can measure the rate of that water uptake by tracking the movement of the air bubble. It is likely that you will be given an experiment where the conditions of the environment is the independent variable. This is what you change. The investigation that's done here was in 3 different conditions: moist air at 25 degrees, dry air at 15 degrees Celsius, and dry air at 25 degrees Celsius. After the first investigation had been completed, the air bubble moved along the capillary tube. This question asks you to suggest how to move the air bubble back to the 0 point. This is the function of the reservoir of water that you have here. If you turn the tap, you can allow the same amount of water that you lost via transpiration back into the capillary tube, and so the air bubble will return back to the 0 position. So what you need to do here is simply turn the tap and allow the air bubble to go back to the 0 position. This next question concerns the capillary tubing itself. You can see that the oil moves further up the capillary tube when it's more narrow. This question is asking you to explain why a narrow tubing was used. Think about the piece of equipment next to the capillary tube. We are measuring the distance that the air bubble moves. If we used a wider tube, the bubble wouldn't move as far and it will be hard to see, it will be hard to read the movement. So your first mark comes from saying that the air bubble moves further along, and this will improve the accuracy of your reading. This next question gives us a graph showing how the position of the air bubble changed over time for the first investigation. It tells you that the cross-sectional area of the tubing is 0.8 millimeters squared. I've got this diagram here to show you what that means. It's just the area of a flat 2D circle that fits inside the tube. From this we can calculate the rate of water uptake for the first investigation. Since this is a straight line, we can use any point along our line, and the easiest to use looks like this one here, where the two bold lines meet. That corresponds to 3 minutes on our x-axis and 40 millimeters on our y-axis. So 40 millimeters over 3 minutes gives us 13.3 millimeters per minute. This is the rate that the air bubble moves. But now that we know that, what we need to simply do is multiply that cross-sectional area by the rate that the air bubble moved to work out the rate of water moved, and this gives us 10.6 millimeters cubed per minute. To prepare you for the last question, have a go at these challenge questions here. Sunlight and plants will always be about photosynthesis. So whenever you see sunlight, whenever you see plants together, photosynthesis, my G. And remember that this is the equation. Carbon dioxide plus water gives us the carbohydrate, glucose and oxygen gas. So carbon dioxide needs to enter through the stomata to allow photosynthesis to happen. Transpiration is the loss of water vapor through the stomata. Now the reason why a dark humid location would reduce transpiration is because firstly the stomata open when the sun is shining. So in a dark location the stomata are closed due to no photosynthesis, but also the concentration gradient for the water is low due to high humidity. Check out my video on diffusion and active transport for more information on concentration gradients. But please don't acquire this misconception. Water enters through the roots and not through the leaves. It leaves through the leaves.
I am leaving!
And so we have this question here, asking us to explain why the air bubble didn't move in the dark. You know what to do now. The stomata are closed because no photosynthesis can happen, and so no transpiration can happen because the stomatas are closed. And look at that, the lesson is done.
Fatality.
If you stuck around to the end of this video,
I appreciate that.
check out the timestamps to go back to any part of this lesson. Check the descriptions to chop some of the goodies, and I'll catch you guys in a bit.